Today, we're talking about a molecule you think you know inside and out, glucose. Yeah, that simple sugar that fuels us. But trust me, its life is anything but simple. We're going to peel back the layers and look at the secret life of this little guy. How this simple chain of atoms twists, connects, and literally builds the world around us.
So, when I say glucose, what do you picture? Come on, be honest. If you're anything like me, there's probably one specific image from a high school textbook just seared into your memory.
Yeah, something like this, right? This straight, open chain. It's called a Fischer projection. And look, it's not wrong. It's a totally fine way to draw glucose. It's simple, it's clean, you see all the carbon atoms lined up in a row. Easy peasy.
But, and this is a huge but, in the real world, inside our bodies, in that watery soup of our cells, glucose almost never looks like this. That straight line, it's just a blink and you'll miss it moment in a much, much more interesting story.
You see, glucose is kind of like a natural-born contortionist. It doesn't want to stay stretched out. It'd much rather fold up on itself, twisting from that straight line into a comfy, stable ring. So, how does it do it?
Okay, so here's the key. You've got this reactive part, the aldehyde group, right at the top on carbon number 1. Well, it gets a little grabby. It reaches all the way around and links up with the hydroxyl group on carbon number 5. And when that happens, snap! The whole chain curls up and closes into what we call a hemiacetal ring. Think of it like the molecule just getting into a much more relaxed, stable position.
But here's where it gets cool. This simple act of closing up creates a brand new situation. See that new hydroxyl group that just formed on the first carbon? Well, it has a choice. It can either point down or it can point up. And depending on which way it goes, we get two slightly different flavors of the glucose ring. We call these anomers, and they get their own names, alpha and beta.
So how do we even know they're truly different? I mean, they look almost identical, right? Well, it turns out we have a clever way to see the difference. We can shine a special kind of polarized light through a solution of pure alpha glucose. And when we do, it bends that light by a very precise amount, a positive 112.2 degrees.
Okay, now hold that thought. What happens if we do the exact same experiment, but with pure beta-glucose? We get a totally different result. It only bends the light by positive 18.7 degrees. This is the smoking gun. It proves they are two distinct molecules with their own unique properties, even though they're built from the exact same parts.
Okay, but this is where the real magic happens. Let's say you take either the pure alpha or the pure beta form and dissolve it in water. You'd expect the number to stay the same, right? Wrong. The reading starts to drift. It changes until it finally lands on a stable, final value of positive 52.7 degrees. This amazing process is called mutarotation, and it's the ultimate proof that these rings aren't static. They're constantly popping open back into that straight chain and then snapping shut again, flipping back and forth between their alpha and beta selves. So what we end up with is this stable mix, an equilibrium. In water, at any given time, glucose is mostly the beta form. That's the most stable, making up about 63%. Then you've got the alpha form at around 36%. And what about that straight chain we first talked about? It's there, but it's less than 1% of the population. It's super rare, and this is crucial, that tiny, tiny fraction is the secret to some of glucose's coolest chemical tricks. And that's our perfect segue. That incredibly rare open-chain form with its exposed aldehyde group is exactly what gives glucose its superpower. It's what makes it something called a reducing sugar, and it's the key that unlocks a whole world of important chemical reactions.
So, what on earth is a reducing sugar? It sounds complicated, but the idea is simple. It's just a sugar that's generous. It can donate electrons to another chemical. And for glucose, that generosity comes from one place and one place only, that aldehyde group that's briefly exposed every time the ring pops open.
Now, if you put it in the right environment, like an alkaline solution, that open-chain glucose can shapeshift into an even more reactive form called an ini-diol. You can think of this ini-diol as an electron-donating powerhouse. It's just itching to give away its electrons. This is the real engine behind glucose's reducing power.
And we can actually see this happen with our own eyes in a couple of classic lab tests. You take your sugar and add it to this bright blue liquid that has copper ions in it. If your sugar is a reducing sugar, that enediol form gets to work, donating its electrons to the copper. This changes the copper, reducing it.
And here's the cool part. The new copper can't stay dissolved, so it literally falls out of the solution as this awesome brick red solid. It's a fantastic visual yes or no test for that reducing power.
But wait, there's more. This reactivity isn't a one-trick pony. It lets glucose and its cousins do all sorts of other things. They can be oxidized into sugar acids, or they can be reduced to make sugar alcohols like sorbitol. They can even react with certain chemicals to form these really beautiful, distinctive crystals called osazones. Back in the day, this was a huge deal for chemists trying to figure out which sugar they were looking at. Okay, so we've seen glucose twist itself up, and we've seen it give away electrons. But its next trick, well, this one might be the most important one for life itself, and that's its ability to connect with other molecules. The magic behind this is something called the glycosidic bond.
So what is a glycosidic bond? Remember that special hydroxyl group on the first carbon, the one that made our alpha and beta forms? Well, when that group reacts with another molecule, it forms a link, a covalent bond, locking them together. This single type of bond is responsible for building pretty much every complex carbohydrate you can think of, from the sugar on your table to the starch in your potatoes.
And get this, it's not just about linking sugars to each other. This bond connects sugars to all kinds of other things. I mean, just look at this stuff. That amazing vanilla flavor, that's a glycoside. The life-saving heart medication, digoxin, that's a sugar attached to another molecule. Powerful antibiotics like streptomycin, even famous poisons used in research, they all rely on linking a sugar to something else. This bond is absolutely everywhere.
So all this ability to react and connect, it means that our humble little glucose molecule is basically the head of a massive, incredibly important chemical family. We call them monosaccharide derivatives.
Think of the basic sugar structure as a Lego brick. By making just tiny little tweaks, swapping one piece for another, adding a little bit here, biology creates this huge toolkit of specialized molecules. You get sugar acids, you get amino sugars, all these things that are the fundamental building blocks for stuff like your cartilage, or even the cell walls of bacteria.
And that brings us to the ultimate derivative, the biggest one of all. You see, the very backbone of our genetic code, of DNA, is made from a sugar. The D in DNA, it stands for deoxyribose. That's a deoxy sugar, just a sugar that's had one little oxygen atom plucked off. Let that sink in. The entire blueprint for who you are is written on a framework built from a modified sugar.
So the next time you hear the word sugar, I hope you don't just see that flat, boring line from a textbook. I hope you see a dynamic, shape-shifting ring, a powerful little chemical engine, and the literal backbone of life itself. It really goes to show you, right? Sometimes the simplest things hide the most incredible secrets. Makes you wonder, what else are they hiding?